Welcome to BioLearn, where you will learn biology at your own pace. Our lesson for today will center on gene expression. Now, just to give you an overview of the gene expression, remember during our discussion of the central dogma of molecular biology, we learned that it includes the process of DNA replication, transcription, and translation. You may watch the video I prepared about the DNA structure and DNA replication, and I suggest that you watch that first before watching this video. Okay, so as you can see here, for the gene expression, we have here the process of transcription and we have here the process of translation. Now, let's say that this is your DNA strand. Okay, and we have here the template strand, which is the 3' to 5' direction. Again, our DNA template strand is always the 3' to 5' direction. And we have here the complementary strand or the non-template strand, which is the 5' to 3' direction. Now, the process of creating an mRNA or an RNA from the DNA template strand, we call it transcription. Now, the process of creating a protein using the mRNA, we call that translation. Now, let's proceed. Let's study each of the process in detail. But before that, let us first differentiate the difference between a DNA and RNA. Okay, so DNA will proceed to the basic differences between them. All right. So for DNA, one of the differences that it is double-stranded. Okay. Again, we have here the double-stranded DNA, while the RNA is a single-stranded structure. Okay. For the DNA, the nitrogenous bases are adenine, cytosine, guanine, and thymine. Okay. While for RNA, we have cytosine, guanine, adenine, and uracil. Now, let's talk about the complementary base pairing. For DNA, adenine bind with thymine, cytosine binds with guanine. While for RNA, adenine bonds with uracil because thymine is not present in RNA, and cytosine binds with guanine. Okay, now let's be specific with RNA. The three main types of RNA are the following. We have the messenger RNA, the transfer RNA, and ribosomal RNA. Now, I will not be discussing yet the difference between each of the types of RNA, but we will be mentioning this in the process. So later, you will appreciate the difference among the three types of RNA here. Now let's start with transcription. Now to give you an overview of how it happens, let's watch this short video from YouTube.
The basic unit of all living tissue. In most human cells, there is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus. The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
Right? So that already summarizes the process of transcription and translation. But today, let's go to each of the process in detail. Let's start with the process of transcription. Again, transcription is the synthesis of RNA using the information in the DNA, specifically the template strand of the DNA. So it happens inside the nucleus for prokaryotes, and it happens in the cytoplasm for prokaryotes because we do not have nucleus in prokaryotic cell. Okay, so if you will look at this picture here, let's say this, we have here the bacterial cell or the prokaryotic cell. So we have here the DNA, and the template strand of the DNA serves as a template in creating an mRNA. Okay, now for this one, for eukaryotic cell, you have here your DNA, which serves as a template in creating your RNA. Okay, but as you can see here, we have the pre-mRNA and the actual mRNA, because in eukaryotic cell, we have this what we call modification. Okay, so in eukaryotic cell, the mRNA has to be modified in some sort first in order for it to be used during the process of translation. So in short, the process of transcription in eukaryotic cells are a little bit more complicated than the process of transcription for prokaryotic cell. Okay? So the transcription occurs in the 5' to 3' direction. Okay, just like how DNA replication happens, no? And the RNA transcript is complementary to the DNA template strand. So what does that mean? Okay, so this is the DNA template strand that's 3' to 5' direction. And this will serve as a template in creating the mRNA. And the mRNA will be formed from 5', look at this, 5' to 3' direction. And as you can see, this is anti-parallel to the DNA template strand. So adenine binds with uracil, cytosine bonds with guanine, cytosine bonds with guanine, adenine bonds with uracil, and so on and so forth. Okay. What happens in translation later on, we will discuss that. But basically, just like with DNA replication, in transcription, we also have 3 stages. It's the initiation, elongation, and termination. Let's start with initiation. So in initiation, basically, that's why it's called initiation, because this is the first part of the process. Okay, and we have here the RNA polymerase, which is the enzyme that is responsible for catalyzing the synthesis of RNA from a DNA template. Okay, so we have here the RNA polymerase, and this is the enzyme that binds to the promoter sequence. Okay, so promoter sequence or specific DNA sequence where the RNA polymerase attaches. So it's like the origins of replication in the process of DNA replication. Now, look at this. Initiation. After RNA polymerase binds to the promoter, the DNA strands unwind, just like where is my cursor here. So it unwinds, and the polymerase initiates RNA synthesis at the start point on the template strand. So again, RNA polymerase attaches to the promoter sequence, the DNA sequence, or the DNA unwinds, and the RNA starts to form, specifically mRNA. Okay. Now, in the process of elongation, the RNA polymerase reads the template strand from 3' to 5' direction. Okay, so basically, this is the template strand, itong nasa baba, kasi that's the 3' to 5' direction. So if the RNA polymerase reads the template strand from 3' to 5', it means that the movement of this RNA polymerase here is from left to right. Okay, as per, ano, in respect to this diagram here. Okay, now, since the complementary strand or the RNA is also anti-parallel to the DNA template strand, what is forming here is a 5' to 3' direction of RNA, and we call it elongation. So here, the RNA transcript carries the same information as the main template strand or the coding strand or the complementary strand, but it contains the base uracil instead of thymine. Ano ibig sabihin nun? So if in the template strand you have the adenine, then the RNA which will form here is uracil. Thymine, cytosine. Guanine, cytosine. Thymine, adenine. Okay, so that's it happens. Elongation because the RNA design elongates. Okay, terminate here, look at this. So as you can see here, let's say this is the template strand, right? This is the 3' to 5' direction. What is forming is an mRNA that is in the direction of 5' to here. 5' to 3' direction. Here in this diagram, this is the template strand, 3' to 5' direction. And this is your mRNA, 5' to 3' direction. As simple as that. Okay. Now, for prokaryotic cell, that mRNA is already okay to undergo translation. But for eukaryotic cell, it has to undergo muna modification, which we will discuss later on. But before we end that process, let's now proceed to the process of termination. So in the process of termination, it simply means that this RNA polymerase will now reach the terminator sequence. If you have the promoter sequence, we also have the terminator sequence. And on that terminator sequence, what will happen is that the RNA transcript will now be released. And that is now your mRNA. And again, that mRNA in bacteria will now be ready for translation. However, for eukaryotic cell, it has to undergo modification first. Now let's talk about the modification of the pre-mRNA after transcription for eukaryotes. Okay. By the way, why do we have to modify them first? Una, kasi later we'll discuss that. Pero let's talk muna about the modification that happens here. The first modification is the alteration of the pre-mRNA ends. Okay. Here, no, the 5' end is now, it receives a 5' cap. Ano yun? It's a modified form of guanine nucleotide with 3 phosphate groups, no? So it's made up of 3 phosphate groups and guanine bases. And for the 3' end, this is now the poly-A tail because it is made up of many adenine molecules. Okay? So, para siyang sapatos ng mRNA. Later on, this is actually for the protection. Okay? So, ano na ulit yung kahalagahan ng dalawang yun? Number one, it facilitates export of the mature mRNA from the nucleus para makalabas siya from the nucleus outside, papuntang cytoplasm. Number two, it helps protect the mRNA from degradation by hydrolytic enzymes. So it serves as a protection from hydrolytic enzymes. And next, it helps ribosomes attach to the 5' end of the mRNA once the mRNA reaches the cytoplasm. Because during the process of translation, or the process of translation will start in the 5' end here. Okay, so that 5' cap will help attach the ribosomes on the 5' end here to start translation later. The second modification is the RNA splicing. So what happens during RNA splicing is that, remember, your DNA is made up of introns and exons. When we say introns, it does not code for any amino acids. If it's exons, it is translated into amino acid sequence. So the goal of RNA splicing is to remove the introns. What will remove the introns? We call it spliceosome. Okay? It's a protein known as spliceosome which will breakdown introns. Ano na lang matitira? Ang mga matitira na lang ay exons. So this is now the modified version of the mRNA, and this is now ready for translation. Let's now proceed to the process of translation. Okay? So here in the process of translation, we simply translate mRNA to protein. And we also have 3 steps: initiation, elongation, and termination. Okay. In the process of translation, please take note that the transfer RNA translates mRNA to amino acids. Okay, it's the transfer RNA that translates mRNA to amino acids. Ano yung transfer RNA na yun? This is transfer RNA. So ano na yung mga nabanggit natin? We have the mRNA, and this time this is the tRNA. So this will help in translating the mRNA to amino acids or proteins later. Okay, and please be reminded of this codon chart also. Okay, so if yung mRNA ay UUU, then it will be translated into phenylalanine. This is an example of an amino acid. UUA, leucine, UUG, leucine, and so on and so forth. Okay, so let's try this one. So let's say this is your mRNA sequence, no, that was made during the process of transcription. So let's say you have here the AUG. No? By the way, each 3 bases, nucleotide bases, we call them codon. Okay, so we call them codon. They are the nucleotide triplets. So let's say we have here the AUG. AUG is translated into methionine. And please take note that AUG is also the start codon. Okay, so dito lagi nagsisimula ang translation. So hanggat wala pang AUG, hindi pa magsisimula. So AUG will be translated to methionine. AUC will be translated into here, ILE. Okay. Then UCG, serotonin, here. And then UAA, stop. Kapag namit niya na yung stop, end na ng translation. So ito na ngayon yung ating amino, ito na ngayon yung ating protein. Okay. So amino acid 1, amino acid 2, amino acid 3, then the stop codon, then here, this is now your polypeptide or your protein. Ano naman ang role ng ribosome? Okay. Ang role naman ng ribosome, this is where the actual protein synthesis happen. So, isa-isahin natin ito. Now, in your ribosome, it's made up of the bigger subunit, and you have here the smaller subunit. And then it's made up of the E side, the P side, and the A side. Here, this is where in the P side or the peptidyl tRNA binding side, it holds the tRNA carrying the growing polypeptide. So this is where the amino acid elongates. In the E side, this is where it exits. The tRNA used in the P side will now exit the E site. And the A site will, or the amino acid site, will hold the tRNA carrying the next amino acid to be added to the chain. So later on, we'll discuss it further. Okay, let's watch this video.
Initiation.
So you have here the ribosome.
The
This is
small
bigger
ribosomal
unit and the
subunit
smaller
binds to the
subunit.
initiator tRNA
Okay.
carrying the
Now,
initiator
so we
amino
have here
acid
the
methionine.
tRNA, and then as you can see here, it brings
This
an amino
complex then
acid
attaches to the
methionine.
cap
So
structure
kanina
at the
ito,
5' ends of an
kanina ito dapat,
mRNA
itong
and scans
tRNA
for
na
the
may
start
dalang
codon
methionine, dapat
AUG.
mahanap niya si AUG,
The
which
process
happens
is
to be
mediated
the
by
start
several
codon.
initiation factors. At the start codon,
So as
the
soon
large
as the
ribosomal
tRNA
subunit
binds to
joins
the
the
AUG,
complex and all
you have now
initiation factors
methionine.
are released.
Okay, it
The
will now,
ribosome
okay,
has
so
3
this is the
sites.
E
The A
site.
site is the
Yeah,
entry
A
site
site.
for new
This is where
tRNA
the next
charged with
tRNA
amino
will
acid
enter.
or amino acyl tRNA.
So it will
The
look
P
na,
site is
yeah, P
occupied
site.
by
This is
peptidyl
where
tRNA,
this
the
amino acid will
tRNA that
elongate.
carries the growing polypeptide chain. The E site is the
And
exit
the E
site
site,
for the
this is where the
tRNA after it
tRNA
is done
used here
delivering the
in the
amino
A
acid.
site and P site. This is where it will
The
exit.
initiator
The
tRNA
ribosome.
is positioned in the P site. Elongation.
Now,
A new
so
tRNA
UUU,
carrying an amino acid enters
is
the
it
A
okay?
site
Is
of
it
the ribosome.
complementary to UGG?
On the
No. What is
ribosome,
complementary to
the
UUG
anticodon
is
of the incoming tRNA is
Now,
matched
we
against
have
the
to wait
mRNA
for a
codon positioned in the
tRNA
A site.
carrying that anticodon AAK
During
to
this
enter this.
proofreading, tRNA with incorrect anticodons are rejected and replaced by new tRNA that are again checked. When the right amino acyl tRNA enters the A site,
All right, so as you
a
can see
peptide
here, a
bond is made
tRNA
between the two
carrying an
now
anticodon
adjacent amino
AAA is now
acids.
available and ready to enter the amino
As the
acyl site.
peptide bond is formed, the tRNA in the P site
Okay, so your
releases the
ribosome
amino
will not
acids
move
onto the
until it
tRNA in
reaches
the
the
A site
P site.
and becomes
Okay,
empty.
and then as you can see here, the
At the
used
same
tRNA
time,
will now
the
proceed
ribosome
to the E
moves
site.
one triplet forward on the mRNA. As a result, the empty tRNA is now in the E site and the
And the
peptidyl
same process
tRNA
will
is in the
happen.
P site. The A site is now unoccupied and is ready to
There,
accept a
it's
new
now going to
tRNA.
exit the ribosome.
The cycle is repeated for each codon on the mRNA.
CCG there. Oh, I want you to watch the video. Anyway, thanks to I obtained this video from Alila Medical Clinic
Termination.
or Medical Media
Termination
from YouTube.
happens when
Now,
one
as
of the
soon as
three
this
stop
ribosome
codons
reaches
is
the
positioned in
UH,
the A site.
specifically the A site, no?
No tRNA can fit in the A
A
site at
release
that
factor,
point,
which is a
as there are no
protein, will enter
tRNA
the
that
A
match
site.
that sequence.
Okay, so it will now enter
Instead,
the A site.
these codons are recognized by a protein, a release factor.
And as you can see,
Binding of the
the
release
amino
factor
acid
catalyzes the
are now
cleavage of the
elongated.
bonds between the polypeptide and the tRNA.
There, and as soon as release
The
factor attaches to the
polypeptide is
UAG,
released from the
it will now
ribosome.
release the
The
amino acid which
ribosome
was
is
formed
dissociated into
in the
subunits
process earlier.
and is ready for a new.
Okay, now, so translation also has 3 stages. We have the initiation, elongation, and termination. So during the initiation stage of translation, it brings together the mRNA, a tRNA bearing the first amino acid, and the 2 subunits of the ribosome. So you have here the 2 subunits of the ribosome. All right. Now, during the process of elongation, the ribosome now will move until the amino acid elongates. Okay, so it gets longer. During the process of termination, a release factor will now enter the A site, where the A site is now on the UAG codon. Okay? And you have now the amino acid. Okay, you have now the amino acid. This will now fold to be used as a protein. Okay? So remember, the end product of the central dogma of molecular biology is protein. And this is now protein. How it becomes a protein? It starts with DNA replication, followed by transcription and translation. So that ends the discussion on the gene expression. Thank you.